Biodiversity, encompassing the variety and variability of all life on Earth, is the product of over 3.5 billion years of evolutionary history. An estimated 8 to 14 million different species are believed to inhabit the Earth. To date, only about 1.7 million species of animals and plants have been described, while many more await discovery. Forests, with all their varied types, are the single most important repositories of terrestrial biological diversity. Forest genetic resources constitute a very important subset of biodiversity. Conserving forest genetic resources, FGRs, are vital as they are unique and irreplaceable resources for the future. In India alone, more than 340 million people are estimated to be dependent upon the FGRs for their livelihoods. As per present state of knowledge, 18,236 higher plant species have been documented from India so far. More than 80% of this higher plant diversity is contained in the forest habitats. About half of this forest plant diversity constitutes FGRs. To generate understanding and knowledge on forest genetic resources and to develop and strengthen their in situ and ex situ conservation programs, Forest Research Institute has initiated a program on documentation, characterization, storage, and conservation of these valuable resources. Various steps have been taken to safeguard this nature's treasure. The first and the foremost requirement for management of a resource is comprehensive documentation of the resource. A total of 250 species have been shortlisted. That includes 141 tree species, 29 shrubs, 15 lianas or woody climbers, and 65 rare, endangered and threatened species. Those species are being studied in detail with respect to their diversity, regeneration, population structure. Field survey of all districts of Uttarakhand has been carried out for ground verification of existence of species and their populations. During this survey, several species were enumerated and their regeneration status has been studied. Quantitative analysis of vegetation is also being done. A total of 50 FGR species were shortlisted for the development of eco-distribution maps. Eco-distribution mapping of species is carried presently with latest geospatial modeling tools, ArcGIS and ERDS Imagine software, besides using Maxent species distribution model. Landsat 8 satellite image of Uttarakhand was prepared for this mapping. The DD Herbarium of FRI is the second biggest herbarium in the Indian subcontinent. It was established in 1906 and contains 330,000 specimens. For the better access and long-term safeguard of specimens, the herbarium has been renovated and the digitization work of the specimens has been undertaken. Protocols are being developed for keeping the seeds in short, medium, and long-term storage in collaboration with the National Bureau of Plant Genetic Resources. Surveys have been conducted for demarcation of populations and availability of their seeds. Seeds of 100 important FGR species will be kept for long-term storage and conservation. So far, the well-processed, cleaned, desiccated, initial viability determined seed samples of 50 forestry species have been deposited in the gene bank of NBPGR New Delhi for their long-term conservation. For germplasm of very high conservation concern and those having recalcitrant seeds, in vitro storage technique is being adopted and their protocols have been developed along with the red listed species of FGR in the form of pollens. Forest genetic diversity, both interspecific and intraspecific, serves a number of fundamentally important functions. 6 species have been prioritized for molecular characterization and genetic diversity estimation. These are Rhododendron arboreum called Buras, Texas wallichiana called Thuna, Quercus semicarpifolia called Kharsu oak, Betula utilis called Bhojpatra, Myrica esculenta called Kafal, Diplonema butyracea called butter tree. Extensive survey and sampling work have been initiated in Uttarakhand for the selected species. The samples of these populations are segregated for chemical examination and DNA fingerprinting. This would help in identification of regions with high level of genetic diversity and private alleles which are most suitable for conservation. The habitats of many FGRs, especially the critical ecological niches, are experiencing an increasing pressure leading to further degradation and fragmentation, causing further depletion in the populations. In situ and ex situ conservation measures for such species in the form of conservation reserves and the field gene banks are being developed. 5 priority species have been shortlisted for this purpose. These species are Cinnamomum tamala, Diplonema butyracea, Rhododendron arboreum, Myrica esculenta, and Texas wallichiana. Nursery techniques of these species have been standardized, and planting stock of priority species has been raised for establishment of field gene banks. Evolved over millennia in their natural habitats, the forest genetic resources have a huge potential in ensuring food and health security of the country's burgeoning human population and its livestock. It is likely that many of these species might disappear even before they are explored and described or assessed for their potential value to the humanity. This program is a comprehensive step towards conservation, development, and sustainable utilization of FGRs for their benefits to humanity.